All right, so I'm going to give you an introductory to what the kidney does as far as its functions, and then some of the aspects of development of that, and then some of the things that we're kind of exploring today in development regarding how to build a new kidney, as they've started to show that they can actually start build a new kidney from stem cells, but it has very limited capacity. And they've only done it in mice and some whatnot. But that would be a huge, huge thing if we could actually build new kidneys from stem cells for therapeutic use for people who have renal failure. Okay, so the intermediate mesoderm, which is between the paraxial mesoderm and the lateral plate mesoderm, that's what becomes the kidneys and the gonads. But we're saving the gonads for the sex determination lecture, which is everything about how the sex organs essentially develop, how the testes and the ovaries develop, as well as the respective ducts that are with those. So everything today is primarily going to be on the kidney aspect, though there will be a part where you actually see where the gonads are developing in relation to the kidneys developing as well. One of the things that they've shown experimentally is that the paraxial mesoderm is the inductive tissue for the formation of the kidneys. They show that if you separate these two, you do not get kidneys forming. You will not get the intermediate mesoderm undergoing the necessary differentiation that allows the kidneys and the gonads to form. Over here, you're seeing that it's, in fact, they were able to do experiments where they extracted both the intermediate mesoderm and the paraxial mesoderm, put them just right next to one another in vitro, and you still get that inductive signaling. So not only is the paraxial mesoderm necessary, but it's also sufficient. You don't need many other tissues or any other tissues to induce the intermediate mesoderm to become the kidney. And this is actually a good thing, because this gives us hope and promise that we might be able to culture cells and grow a kidney. There's still a lot of processes that we're figuring out, but kidneys are one of the organs that we know quite a bit about how it forms. Other organs are very complex. Kidney's very complex. We're going to go over the complexity, but it's one of those that has one of the greater potentials for therapy in terms of renewing or regenerating a whole kidney for some type of therapy. Now, some of the major genes, we've talked about limb genes before. In this context, there's also PAX genes that are necessary for this. I'm mainly going to show the overall induction processes. I'm not going to require you to know every single gene. They're still figuring out a lot of these, but there are going to be a couple of key genes that I mentioned in the induction process of the kidney. So this just kind of gives you an overview, kind of a topside dorsal view of the paraxial, intermediate, and lateral plate mesoderm. So here in between that, but sandwiched between these, is essentially the intermediate mesoderm. Again, the paraxial mesoderm is both necessary and sufficient, as they've shown through various transplantation and isolation experiments and even some genetic studies, that the inductive signals from the paraxial mesoderm are absolutely necessary, but also sufficient to create the kidney. So let's look at how this forms. One of the main regions of the intermediate mesoderm forms into this duct, which we call the pronephric duct. So the pronephric duct starts forming more anteriorly and then starts regressing backwards. Now, in different organisms, different parts can play slightly different roles. So we're primarily going to focus on human kidney development. But in mice and in other organisms, there are some substantial differences between what degenerates, what forms, what does what, and so on and so forth. So we're primarily going to focus on human kidney development as I go through here. So just know that that's what I'm going to be doing that on. Now, the pronephros, one of the things about the pronephros is even though it degenerates through apoptosis, it's necessary because it induces the more posterior mesenchyme, or intermediate mesoderm, to become the next part of the kidney formation, which is the mesonephros. Okay? So as the pronephros degenerates, it induces the tissue just posterior to it, down the anterior-posterior axis, to start undergoing differentiation, and this becomes the mesonephros. So this is kind of what it looks like. Here we've got, in the red, we've got the intermediate mesoderm. As in the anterior region where the intermediate mesoderm is forming, you get the pronephros forming. You get the pronephric duct right here, and you get these pronephros forming, these protrusions from the pronephric duct. Well, eventually these start degenerating, but there are inductive signals from this that cause the more posterior mesoderm to become the mesonephros. Now, in humans, the mesonephros will also degenerate. It will go away, but there are certain derivatives of the mesonephros that do play a key role. For example, from the mesonephros, you get your hematopoietic stem cells, which are the precursors to everything in your blood. So remember we talked about the bone marrow and limb bones. The stem cells that form all of the red blood cells, white blood cells, platelet, the platelets and whatnot, these come from the stem cells that originate from the mesonephros that will eventually migrate into these limb bones and ultimately form those hematopoietic stem cells. So you don't get these stem cells coming from the lateral plate mesoderm. You get them coming from here, the mesonephros. Another important role is in males. Mesonephros plays an important role because it's forming right next to where the gonads form, plays an important role in the sperm-carrying tubules to form. So those are two derivatives that the mesonephros essentially becomes. It is the main source of the hematopoietic stem cells, which is the stem cell in your bone marrow, and that's where, when you do a bone marrow transplant, these are the stem cells that you're transplanting from one person's bone marrow to another. And in males, the mesonephros also forms the sperm-carrying tubules from the testes. So as the testes form, the mesonephros, certain cells will form these tubes that are necessary for carrying the sperm from the testes into the Wolffian duct, and then as that forms. The rest of the cells essentially degenerate. Okay? So again, just like the pronephros, the mesonephros induces the next set of tissue, and most of that degenerates. But certain cells are derived, as they've shown, from the mesonephros, such as hematopoietic stem cells and these sperm-carrier tubules. They do not, in women, you don't get any type of these tubules forming because of the formation of the ovaries rather than the sperm. And we'll get more into that when we talk about sex differentiation. A lot of that comes down to signals, inductive signals from the gonads, such as FGFs and things of that sort. But that's necessary. It's only in males that you get that inductive signaling, which is why that's why it forms in there. So the main thing we're looking at is what we call the metanephrogenic mesenchyme. So you have the pronephros, which induces the mesonephros. The mesonephros then induces the metanephros, or what we call metanephrogenic mesenchyme, and this is where your kidney forms. This is the material which forms in conjunction with the nephric duct. So as you can see, you get this nephric duct that forms from the anterior to the posterior. Eventually, all of this degenerates except for this most posterior region. So the interplay between the metanephrogenic mesenchyme and the nephric duct, that's what creates the kidney. Now, there are many different cell types in the kidneys, at least 12 different cell types within the nephrons themselves. And this is why these are called the nephric ducts, is because they're necessary to form the nephrons, which are the main functional component of your kidney. Each kidney has about 1 million nephrons, and the nephrons are the filters, essentially, for your blood. Your kidneys will filter about 180 liters of fluid a day, which is about 70 times your blood plasma. This is how often your kidneys have to filter your blood from the metabolic waste and the production of urine and things of that sort. Major role. Now, your kidneys don't just filter your blood. There's actually a number of other things that your kidneys do. They will regulate blood pressure, so they actually send out hormones that can help regulate blood pressure at your heart. They eliminate waste. They also reclaim water. This is one of the reasons why we can live on land and other organisms can't. It's because we can conserve water. There are various hormones that ultimately affect the collecting ducts within the kidney that reabsorb water when we're dehydrated. Otherwise, we'd lose so much water as we're trying to filter out our blood, we would not be able to keep up with it. And it also maintains levels of balance of ions, pH values, and things like that. So kidneys do more than just eliminate waste, regulating blood pressure, maintaining homeostasis with ion concentrations, you know, a lot of different things, reclaiming water in cases of dehydration. That's why when you don't drink a lot of water, your urine is a lot more yellow, is because your body is really concentrating the urine. When you drink a lot of water, it doesn't have to conserve as much, which is why it's going to be a little more clear. All right, so the Metanephros. This is the part of the intermediate mesoderm that is going to become your kidney. Now, there are about 9 steps that we're going to go through on how it's essentially reciprocal induction. This whole process of kidney formation is a reciprocal inductive process between the metanephrogenic mesenchyme and the nephric duct, or the ureteric tubes that are forming as a result. So the ureteric bud here that comes off of the nephric bud, this is going to essentially have kind of this little dance of paracrine factors between the metanephrogenic mesenchyme and the ureteric bud. Okay, so let's go through how this works. This just kind of shows you a picture of the formation. Here we have the ureter. Here's the metanephrogenic mesenchyme surrounding the ureter, and here are the ureteric buds that are starting to form, and these will eventually become 1 million branches of these nephron/collecting ducts that all feed into the ureter, which then feeds into your urinary bladder, which then collects all of the urine necessary for excretion. So, reciprocal induction. We're going to go through all of the steps. So the main reciprocal induction you're getting is the ureteric bud, which comes from the nephric duct, and the metanephrogenic mesenchyme. This is the interaction you're getting in the interplay. This is why this is a big candidate for possibly regenerating this organ, is because we understand so much about the interplay between these two groups of cells. There's still a lot of unknowns, but the more we can understand it, we might be able to eventually create a kidney in vitro from someone's stem cells or from someone else's to be able to then put that kidney in and have it function. They have, I'll get at the end of today's lecture, I'll talk about how they did do that for a mouse, but it was a little different than what you might think. They're still not quite at the point where they can do this process in vitro, but we understand quite a bit about it. Now, one of the things that they found is that by the time the metanephros is being induced by the ureteric bud, it's only competent to form the nephrons of the kidneys. If you put it in any other place and you try to put it under some other type of induction signal, it only knows how to make a nephron. So at this point, the signaling from the paraxial mesoderm has caused these cells to already be in a committed state to be able to receive these inductive signalings. So there isn't a lot of potential in this metanephrogenic mesenchyme to become anything else. It's already committed to become the nephros at this point. So here are all the steps. So let me go through most of these steps here and what's going on. So the first step is the formation of the metanephrogenic mesenchyme. As I mentioned, that comes from here, where the pronephros induces the subsequent mesoderm to become mesonephros, and the mesonephros induces the subsequent mesoderm to become metanephros, and that's that first step. So this has to be induced. Once this is ready to go, then it's ready to start this reciprocal induction. They found that there were Hox genes involved, Wnt signals, other genes like Fox genes and whatnot. So the first thing that happens after that is the first tissue to send out an inductive signal is the metanephrogenic mesenchyme. It signals a glial-derived neurotrophic factor to induce the ureteric bud to start branching out. Okay, so essentially, you see right here, this branching is essentially given by the metanephrogenetic mesenchyme. It releases GDNF, and that causes branching out. And that's really where you're going to get these millions of different formations of all of these nephrons going on. Then, in return, the branching ureteric buds are going to make sure that the metanephrogenetic mesenchyme doesn't undergo apoptosis. They showed that if the ureteric bud does not secrete FGFs and BMPs, then this mesenchyme will die. It'll undergo apoptosis and die. So there has to be then a subsequent inductive event from the ureteric bud that's branching out, and that just makes sure that any mesenchyme that is not surrounding these ureteric buds is going to die off. So it makes sure that only that mesenchyme that is within the influence of the developing ureteric buds is going to be sustained. Other cells that do not receive this inductive signal are going to die off. And again, this is part of the shaping process that forms all of the nephrons. Then you have other inductive signals, Wints, mainly here, from the ureteric bud. And this causes the cells to go from a mesenchyme state to an epithelial state. So initially, all of this mesoderm is in a mesenchyme state. They're loose cells. But as the ureteric bud starts branching off in this loose mesenchyme, then the tissue, the cells around the formation of the ureteric buds, it's going to start causing that tissue or those cells to epithelialize. They'll essentially start forming various cadherins. Those cadherins will then cause them to aggregate and to start forming these nephrons, essentially. They'll first form kind of this ball of epithelial cells, and then they'll eventually develop into the nephron itself. So aggregation, going from a mesenchyme to an epithelial transition. This involves cadherins and laminin and fibronectin and other types of proteins that are necessary for that transition from one state to another. Then subsequent Wnt signalings then cause it to form into kind of a tube. So first it aggregates and forms connective tissue or epithelial tissue. Then it'll actually start forming the nephron that will be inserted into the ureteric bud, and that's what creates the entire nephron itself. So first it aggregates, and then it kind of reshapes itself into the actual nephron, and I'll show you what a nephron looks like. Then in return, the epithelial metanephrogenetic cells then in turn signal for further branching of the ureteric bud. And this just keeps going to cause mass branching that creates the millions of nephrons that are the main functional unit of the kidney. And then in the very end, you get the cells that are forming the nephron differentiating into about 12 different cell types that are necessary for the filtration process and all of the functions I'm going to go through here of what the nephron essentially does. So there's actually several different cells. Some are sensitive to osmotic pressure. Others have proteins in them that will pump out sodium and chloride, and so on and so forth. So at this point, most of the cells haven't fully differentiated, but by the end, then they start differentiating into the respective areas of the nephron, and you keep going until the kidney is fully formed. There's still a lot that we have to learn about this process, but we do know about the initial reciprocal inductive events. They've done quite a number of studies, as you can see, on this back and forth between the two cell types. So here it just shows kind of that overall stepwise process. Here the metanephrogenetic mesenchyme will induce the ureteric bud to start branching. This one then maintains the metanephrogenetic mesenchyme to not undergo apoptosis. Then it will cause it to coalesce and go from a mesenchyme to epithelial transition. Then you get the formation of the actual nephron itself, as you can see here. It inserts itself into what will eventually become the collecting duct, which is where the ureteric buds come off of is the nephric duct, which becomes the ureter as they all collect into the large ureter. And then you get further differentiation and development of all of the areas, the glomerulus, the proximal convoluted tube, the loop of Henle, the distal convoluted tube, and the collecting duct. Those are all the major areas of each nephron. And you've got maybe two major different types of nephrons. They're only slightly different, but their overall function is the same. Some are in the cortex, and others are in the medulla of the kidneys. So here's kind of what it looks like. Now, as you can see here, you've got blood vessels or capillaries that are highly associated with this, because that's the whole point of the nephron, is to filter the blood, is to essentially filter out all of the waste products, reclaim all of the nutrients and any water that it needs, and through osmosis. Even is sensitive. The collecting duct right here is very sensitive to antidiuretic hormone, or ADH, which is secreted by the endocrine system. So let me go just briefly over how a nephron works. Here you can see the overall kidney. You have the medulla and the cortex. These are where you have your nephrons. You've got a high degree of association of blood coming in through these regions. And then here's the ureter, and where all the collecting ducts are essentially going to bring all of the waste, the urea and the uric acid and other components, any drugs that you have in your system or whatnot, all of that gets filtered out. So let me go through some of the basics of how the nephron works. First, your capillaries will essentially dump everything that is a liquid component from your blood plasma, essentially, into the glomerulus. Okay? About 70% of all of that's just going to be instantly reclaimed in this region called the proximal convoluted tube. Okay? So it doesn't put any cells into this. The red blood cells don't pass this barrier, but you got water, you got ions, you got sugars, you got all sorts of stuff that just gets dumped into the glomerulus and gets strained out. It's kind of like straining spaghetti. The spaghetti never leaves the bowl, but everything else comes out through the pores. Well, then it's taking all of that and bringing most of it back in. And then you've got 30% left. Well, what you bring back in is all the sugars and a lot of water and ions and things of that sort. Well, the next part, the loop of Henle, this is where typically osmosis occurs. In the descending loop of Henle, it's sensitive to water coming out. In the ascending loop of Henle, it secretes sodium and chloride. It kind of pumps these ions out into this outer area. That increases the capacity for this area to have a low osmotic pressure, and that's what pulls the water out in the descending loop of Henle. So basically, this is why this loops down so far, is because this area is very hypertonic, and therefore water is going to be pulled out through osmosis in the descending loop of Henle in that surrounding area. Then the remaining fluid that comes through the distal convoluted tube, this is where you have certain proteins that actively pump drugs out of your body, various metabolites and things that you have in your blood. If they haven't already been filtered out, they're going to be actively pushed out in this regard. Here's where you get some final changes in ion concentration of sodium and chloride and potassium and things of that sort, depending upon what the body needs. And then the last part, the collecting duct here, this is the final filtration. This is under control of a hormone called antidiuretic hormone, or ADH, that if your body, it's actually impermeable to water, but if your body needs to pull out more water and concentrate your urine even further down, it'll secrete that hormone. That'll open up these channels, and water will just pull right back out. Again, notice it's in the same area where you have this osmotic pressure, where water pulls out through osmosis because of the hypertonic surrounding in that area. So that's really how your blood gets filtered out, is it just goes through this process of dumping everything in, and then pulling most of it back out, then pulling more water out, changing any concentrations in ions that you need, and then pulling even more water out if you need to concentrate your urine down even further. So this part right here, this comes from the metanephrogenic mesenchyme. The collecting duct, this is the ureteric bud. This is what is branching and causing all these different branches to come from the ureteric bud. All of these essentially flow into the ureter, which come from the nephric duct right here. These all funnel into essentially the urinary bladder. Okay. So that's kind of what this is illustrating. Here's the ureter. These are the million nephrons that are forming from this huge just reciprocal induction process of forming the kidney. Now, they've shown that there is some repair, but typically when you start getting renal failure, it's these cells no longer able to undergo the filtration process. And your kidneys can actually function pretty well even at 20 or 30%. But once it starts getting below that, then you start having some real problems. One of the things that they are able to do with a mouse kidney was they actually stripped it down of everything but the basic connective tissue proteins. So they took out pretty much all the cells. They wiped all the cells out, and all they left was just all of the connective tissue, kind of just a matrix to build upon. Then they used some stem cells and allowed them to fill in the gaps, so to speak. Now, from that, they only got a kidney that functioned at about 5%. So it wasn't great, but it was a start because they were able to then actually use stem cells to make an organ that matched more closely, because they're stem cells from that mouse, an organ that would be able to be transplanted in. So in that situation, let's say you didn't have a donor, you could technically strip down a kidney from all of the cells that have those recognition markers that are rejected by our immune system, and then use some stem cells to regenerate, the nephrons, but again, it's still in its infantile stage. So it wasn't, they didn't actually grow it up in vitro through this whole inductive process. They just stripped down the kidney to its basic proteins and then rebuilt it up through some stem cells, which is amazing in and of itself. But there's still a long ways to go before we can actually grow kidneys and use them for replacement. Typically at this point, if you don't have a kidney transplant, you have to have dialysis that filters your blood and through the machines. I mean, we have these huge machines that do what your kidneys can do biologically very, very easily when they're working right.